Okay, Valerio, the streaming is start.
Okay.
So yesterday we discussed the first two projects, sea stress and scenarios, and then we continued with SmartX, that showed us. I am at home, and of course, qualcuno può aprire? Sorry. Okay. No, I'm so sorry. I was saying that today we discussed about these projects and we explored seagrasses and spongy. Today we will continue with another project on shrimps. So we move from seagrasses to spongy to shrimps, as we promised. And we will have, first of all, an overview of the project exposed by Dr. Costantini. And then we will continue two papers on transcriptomic analysis of the results. The first, provided by our Israeli partners, that we welcome today very warmly, because we had together a great research time, and then continue and concluded with a group of students that will talk about post-larvae. After this, and after the coffee break that I suggest everybody to have at home, we will continue at 11:30 with a round table that I hope will be interesting. It is reserved only to the scientists, and it could be useful to have some common view of the projects. We will complete with the conclusive remarks at 12:30. I take this chance to invite also Dr. Perozzi to our round table, because of course her presence from the ministry will be welcome if she's fine. And so I give the moderator is Elia Flalo, right, Eli? It's fine for you?
Yes.
Okay, so Elia Flalo will be the moderator for this morning, the chairman, and I would say that Maria Costantini may start with the first presentation.
Okay, I just share my screen. Do you see my screen?
Yes.
Okay.
Yes.
So, good morning to everybody, and the title of our project is Induction and Marine Sex Shift Processes Towards Applied Environmental Mariculture Biotechnologies. And first of all, I would like to thank the Italian Ministry of Foreign Affairs International Cooperation for founding this project, and that give us the possibility to work together with the Israeli group. This project is an interdisciplinary project that involves a group from the Ben Gurion University with Professor Amir Sagi and Elia Flalo and their groups, together with the Italian group where Dr. Valerio Zupo is the scientific coordinator, and involved myself and 5 fellowship, Francesca Glaviano, Mirko Mutaripassi, Nadia Rocco, Emanuele Somma, and Tomas Biel. Just to introduce the project, I would like to point out that the sexual shift processes naturally occurs in marine organisms as a strategy to assure maximum reproductive success. However, instances of sexual shift were reported as a result of the exposure, for example, to pollutions, to environmental stress, or as well as ocean acidification, which may produce ecological imbalances and a large economic losses, In addition, sex shift may contribute to the establishment of biotechnologies, for example, for monosex or sterile populations in mariculture industries, for profitable cultures, and reduces environmental risks. An ideal model for such studies is the shrimp Hippolyte inermis, that is extensively studied in the laboratory of Dr. Valerio Zupo in Stazione Zoologica in Ischia, which represent a very important model species. In particular, in marine environment, this shrimp is distributed in Sigrias meadows in various European coastal waters, where it produces a stable population and on the lip of Posidonia oceanica. It has a very peculiar life cycle with 2 reproductive periods and 2 generations. In fact, as you can see from this graph, in April, the spring generation produces males and females. In September, the generation produces only males, and after 1 year, produce females by natural sex shift. A sexual shift from males to females is a naturally occurring process, but it also may be induced in laboratory by the ingestion of specific microalgae. In particular, in the lab of Dr. Zupo, it has been demonstrated that some cocconaceae species are able to induce sex reversal in the shrimp Hippolyte inermis. In particular, if you see to the graph on the right of the slide, when they see to the content of stomach, in April-May there is a great abundance of microalgae, differently from the other month, showing in abundance of female respect to the other month. So, the study of determination of sex in crustaceans is regulated by a single gland, the androgenic gland, and this gland goes in apoptosis, as reported in this slide, when the shrimp are fed with cocconaceae diatoms. And there are at the moment some ongoing experiment in lab of Dr. Zupo in order to identify the compound responsible of these processes. Moreover, obviously having a more precise idea of what are the mechanisms that regulate this process may be important for aquaculture application. In fact, mainly a deep understanding of molecular basis of sex change is needed to address various scientific environmental issues and applicative challenges. So, concerning our project, the main goal is to study the Hippolyte inermis sexual shift through the identification of key genes responsible of sex shift in different developmental stages. If we see more in details our objectives, we are trying during this year to construct transcriptomic libraries of Hippolyte inermis specimen at different phases within its natural occurring or diatom compound induces sexual shift. We try to identify genes related to sex reversal and programmed cell death of gonads. This with the aim to identify genes related to the control of sexual shift in crustacean decapods for biotechnological application in aquaculture and medicine. So, in this slide, I reported just the steps that we followed during our project that started with the collection of animals, their dissection, RNA extraction, and construction of transcriptomic library by de novo gene. In particular, concerning the collection, here reported the developmental stages that were collected for our analysis according to the different generation observed in the field. In particular, we collected the immatures, males, young females, and mature females, each of ones with technical replicates. Concerning the sampling strategy, we follow with the appearance of the new generation of the 2 generations, sampling in the field in the spring and summer, corresponding to the 2 periods of year that are reported with the red circle. So then the animal were dissected, and before this they were anesthetized on ice, and we dissected the body and then the eyes. Why we take these two different samples? Because in the body contain gonads and androgenic gland, whereas the eyes are located the X organ sinus gland complex. Then, in the laboratory, we collected other samples at the stage of post-larvae, and we collected post-larvae 5 days old that were fed in 2 conditions: with base food in order to have expected young males, and with base food plus the diatom coconase in order to have expected females sex reversed. Then we, in the meanwhile, we also set up a protocol to extract high quality RNA from all the samples that I showed before in order to have quantity, quality, and integrity useful for RNA sequencing analysis. Then the RNA were sent to Novogene for library construction and transcriptomes. So what are our main results? In this my presentation, I don't give your results because the details will be given in the next presentation. But I would like just to give you the more striking results reached in our project. And concerning the samples, firstly, this is the website that it has been constructed in which all the data from the transcriptome are available. Then, concerning the samples collected in the field, the results very clearly demonstrated that there is a clear separation between the 4 developmental stages analyzed collected in the field, and mainly evident when we see to the eyes that are reported in the right part of the slide. And this is very clear when we see to the heat map of variation of gene expression. For example, here reported in the case of eyes, the variation of gene pattern for females and males. And it's very evident that there is a different gene pattern in which the genes that are down-regulated in females, in red, are green, so means that they are up-regulated in males. Concerning the samples in the laboratory, I would just to show this heat map in which very clear is present the different gene pattern expression between the post-larvae that are fed without diatoms and the post-larvae that were fed with the diatoms and base food. So, as you could see from the following presentation of the Israeli group and our fellowship that belong to the Italian group, we obtained very exciting results from a very exciting project. And I would like just to point the attention on the fact that there was a very close collaboration between the 2 groups, with the 2 visits here in Naples by the Australian colleagues who collaborated with us during the dissection phase of the animals and the development of the RNA extraction protocol for RNA sequencing. Moreover, just in February of this year, we went in Israel as soon as the transcriptomic data were available, to discuss with our colleague about the data and organize the data analysis. Other visits were planned, but due to the pandemic was not possible. So we have just a little delay due to the COVID, but as you will see in more detail in later presentation of our results, we are now able to prepare manuscript and publish the data. So thank you very much for the attention, and I pass the word to my colleague for the presentation of the details on our results. Thank you.
Maria, stop sharing.
Yes, I'm doing. Okay.
Professor Aflalo, are you ready?
Yes, I'm ready. Any questions to Maria for her great presentation or introduction into our more detailed lectures?
In the meantime, any question arrives, I would take this chance to stress once again that that really the cooperation with our Israeli partners was a privilege, and that really this was just a step of further research, because we have collected so many data. How many genes, Eli? I don't remember, but
I'll show some
okay,
of them
okay.
It's
an amount of data that it will be hard to compute them all. But the first results are really very exciting. If there are no other joints, additions, I think, Eli, you can start.
Okay, so I'm going to share. Do you see, just a second, I'll look at screen number. Okay. Could you see the lecture now, or the presentation? Yeah. Okay. So I'm going to present again more detailed about our part of the work, actually following the analysis of the transcriptomic library. So in general, again, we are looking for some important processes related to sex shift or sex change in the crustacean. In our unique model that we harnessed to this task was, of course, the Hippolyte inermis, which is found to be a great model for our work. Again, our main goal is to search for key responsible genes that actually responsible for sexual shift or sexual changes, and of course, following their related cascades in the reproduction system or in degeneration of the male part, since we are dealing with hermaphrodite species. Again, selected genes that will be found interesting will be tested for their effect or their functional in sexual differentiation processes, and even more than that, they will be traced or studied in other aquaculture important species, like the commercially cultured Macrobrachum rosenbergi, the freshwater prawn. If you go into the specific objectives, one of the major objectives for this collaborative work is to generate a wide transcriptomic library of Hippolyte inermis. And for that, we selected a distinct naturally occurring phases and also induced sexual shift, as you can see later on, or hear later on in the next presentation. The aim of this transcriptomic library construction is to identify genes that related to the control of sexual shift and gonad substitution. And additional goals, of course, is to follow or to perform some functional analysis, functional genomic assays on several selected genes related to this sexual shift. And of course, later on, to see if those genes have any important or any role in other commercial or other decapod crustaceans. Okay, I'm going to go in a little bit into the transcriptomic analysis, and for this transcriptomic analysis, we used, again, as we saw from Maria's presentation, a distinct phases or distinct stages in the life or the reproductive cycle of Hippolyte inermis, and all the data that was gathered, again, served to construct the library. We actually sampled animals, sampled the shrimp in the northwest side of Ischia, where there is good populations of Hippolyte inermis in the Gulf of Naples. Two distinct periods of sampling were September and April, and this is due to the unique life cycle or life history of those shrimp. And you can see that those shrimps have two reproductive cycles. One of them starts at April, the second starts at September, and they are different in their nature, as Maria described. In September, we can see that we have selected several distinct phases like immature males and young females, and in April, we selected mostly large females. Again, due to the complexity of generating a transcriptomic library, we needed large amount of RNA. This was achieved by pulling a lot of animals into several replicates. We used 3 replicates from each stage, and the distinct stages are immature males, young females, and mature males. From those pooled replicates, RNA was extracted and sent to Novogen company to the deep sequencing, and the sequences were then sent to us back for the analysis of the transcriptomic library. The transcriptomic library yielded a lot, many genes that are differentially expressed. And you can see here an example of 2 heat maps showing the hierarchical clustering of differentially expressed genes. Actually, this is showing differences in between 2 stages, for example, between immatures and males, or males and females. And you can see that separately in body of Hippolyte inermis or in the eye stalk, you can see we have managed to pull thousands of differentially expressed genes. This overwhelming amount of data is very hard or very difficult to manage or to trace differences that are really important to one of the stages. For this reason, we further went through additional analysis called binary expression patterning. And this patterning, we defined several parameters. The parameters showing that we are setting high or low reads or low expression depending on the read quantity. So many reads defined as high, as 1. Low reads defined as 0. And the parameter is that we have at least 500 reads in order to call this expression as a high. And we need that the differences between high and low should be at least of a fold change of 1.3, and with a p-value of at least 0.05. Here I gave you an example of 2 different binary expression patterns, meaning that we have very high at the immature part or the immature stage, very low at the male, very low at the young female, and again very high at mature female. So this pattern we'll designate it as 1001. On the other hand, we can see different expression patterns showing low at the immature, high at the males, high at the young females, and very low at the mature female, and this would be designated as 0110. And if you look at such binary patterning, and I represent here only some of the patterns which we believe that are very important or very crucial in the sex differentiation process. And you can see here several of them. Here you can see a pattern of 1000, actually representing high expression or differentially expressed genes that were high, very high at the immature and very low in all the other stages. The second one show very high expression in the males with very low expression in all the other stages. And another example, very low at all the stages, but high at the mature females. And this analysis actually gave us more focused view on genes that we can actually attribute their role in a specific developmental or reproductive stage. One of the genes, an example that I can show here, one of the genes that is related or found in the differentially expressed genes patterning was the female specific gene vitellogenin, the yolk protein genes that actually responsible for the production of the yolk in the developing eggs. And you can see here that there is a high homology of the structure of the genes with several domains that if we compare it to other hermaphrodite species, you can see that there is a high homology like the Pandalus platycerus or even to the Macrobrachium rosenbergi. And again, if you go back into the binary patterning, we can see that vitellogenin and also its receptor falls within this binary pattern. It means that it is very high at the females. You can see here more than 500 reads, very low in all the other stages, also with the receptor and also the vitellogenin itself. In addition to this known gene, we found genes that falls with the same expression pattern, binary pattern, showing very high expression in females and very low expression in all the other stages. And it's not the only one. We found plenty of them. Those are actually the interesting genes that opens future research. We have a lot of data here that we still not completely know what is the role of those genes, but since they are related to a specific developmental or reproductive stage, it opens very important or very interesting questions. Another genes were pulled, and one of them is the major male important gene, as Maria showed in relation to the androgenic gland. The major hormone that produce in the androgenic gland is the insulin-like androgenic gland hormone, or the IAG, which is termed as the IAG switch. It's actually responsible for the shift between maleness and femaleness. And this gene was also pulled, or this gene was also pulled from the library, from the pattern related to males, and you can see that it has the unique feature of insulin-like genes. It has the B chain and the A chain, the C peptide, and of course there's a 3' UTRs at the end of it. But again, it was related to the androgenic gland itself. You can see here a previous work of our collaboration with Valerio Zuppo. We managed to identify histologically the androgenic gland. This is a lateral section of mature male, and you can see here the body part of the male. You can see here the base of the fifth walking legs. This is the head part. This is the tail part. And very close to the base of the fifth walking leg, you can see the sperm duct, and adjacent to it, the androgenic gland, which is the specific site of the production of this unique hormone. And again, if you go back to our binary patterning, we can see that IAG falls exactly with the pattern of 0110. It means that it has high expression in males or zero expression in all the other stages. And again, if you look at the same pattern, we can see that there are several unknown male genes, okay? One of them is presented here, but we have more than 130 like this. So it again opens a lot of questions, a lot of interesting future search or future research on those genes that might be related to the sex or the sex changes in this peculiar hermaphrodite tree. And with this, I'm going to summarize. I just gave you only a really tiny bit of the analysis that we are having now. We have a lot of data. We have a lot of work to do with it, and it opens a lot of questions for the future. But to summarize, we have here in hand the first transcriptomic description of Hippolyte inermis, life history, dependent, actually related to the specific stages of the reproduction in this species. And we can see that the sexual plasticity in this species serve as a model for crustacean sexual differentiation, and it's a very good model for that. We can see that the binary patterning that generated in this analysis might be applied also in other important decapod species, serving to pull out differentially expressed genes with importance to other species. And at present, we are in the process of concluding some of the result into a collaborative publication. And of course, it opens the way for future fascinating opportunities between the two groups. And I want to thank our lab member and also our collaborative members, Dr. Valerio, Dr. Constantini, Dr. Mirko Mutalipasi. It was a pleasure to work with them all, Nadia, and of course, Emanuele, and all other collaborative students that are participating in the project. So I want to thank you all for listening.
Thank you. Questions?
I have a question, if I may.
Please.
Well, thank you. It's really a fascinating work, and it's amazing how you have a system where there's so much to discover, and it's really, I mean, if I ever have an opportunity to have another life, I'll become I want to turn into a marine biologist, because it's really a fascinating work. Well, being a molecular biologist, but for other systems, I really appreciated the bioinformatic analysis of this huge amount of genes that you fished out as differentially expressed. And I was wondering, with this nice little system where you can histologically dissect the different sites so well, are you planning to see if to further discriminate the genes that show the same binary pattern of expression in terms of localization, physical localization in the different tissues, just as a further hint of their possible function?
Okay, so first of all, I think that at the moment that most of them are unknown genes, it would be better to go for actually research aiming at their functional genomics. And when we will have some data about that, it will be, of course, goes hand in hand with the localization of their expression in different tissues.
Okay, thank you. I was just thinking it would be easier to go after the androgenic gland specific. I thought that was your aim, but I realized that with sequencing, it's much easier to go for the sequence analysis. Thanks.
If I can add, Eli, I don't know if it is clear, but the shrimps we are managing in different size and life moments are individuals of about 4-5 millimeters length. That is really small. So while Dr. Aflalo and Dr. Sagi in the past did this important research on insulin-like hormones on larger decapods, where the androgenic gland was more or less visible, in Hippolyte, the images that were shown before by Dr. Costantini, the androgenic gland is something of a few cells that are well immersed into the other tissues. So this section is quite, I would not say impossible, but it's a hard job. And I can assure that even dissecting the parts that were then submitted to this analysis, genomic analysis, transcriptomic analysis, were quite hard, no, to be performed. But the important point, in my view, is that Hippolyte inermis is quite a special model for the studies of sex maturation and sex shifts, because as far as we know, is the only organism now showing such an induction of sex change due to food. That is something still unbelievable for us that are studying it. When we find that we give diatoms and they not only change sex, but change their expression, we are every time not surprised. And a second important point is that these insulin-like hormones that were discovered and managed in their physiology and structure by the groups of Professor Sagi and Professor Aflalo are now found also in these new powerful models. So I think for them, having another model on which the activity can be seen, also in an evolutionary point of view, will open new big streets for several types of studies.
I want to add one thing over what Valeria said. I think it is a very, very important question that was asked. And at this point, with very small animals on one hand, but on the other hand, that they are passing through, the same animal is passing through different stages in life, which is expressing as male or as female, the same individual. So then, if we are going into the approach of selecting the best candidate genes and then going into the path of functional genomics through either RNAi or genome editing, then it will give us a good understanding of the role of each gene. And this is the main goal, I think, of the project in the future. And I hope that we will continue with the group of Valerio Zuppo and with Maria Constantini for many years now, because there is room for several doctorate studies on the basis of this fascinating data set.
Yes, I agree. Thank you.
Okay. So we will continue. If there's no more questions, we can continue with the next presentation by Dr. Mutali-Passi.
Thank you.
Diego, your stage is yours. The stage is yours.
Thank you. I'm going to share my screen.
Okay.
Okay. Can you see?
Yeah.
Okay. The first part was presented by Thomas. That is Thomas is our fellowship. Is working with me on post-larvae and the approach on transcriptomic analysis. Okay.
Hello everyone. So I'm Thomas Viel, and today with Mirko, we are going to present you our result on the EXAD project in collaboration with our Israeli partners and our colleague Nadia Rocco, Francesca Cavano, Emanuele Soma, and our supervisor Valerio Zuppo and Maria Constantini. So the EXAD project focuses on the induction of marine sex shift process toward applied environmental and mariculture biotechnology, and the industry Hippolyte inermis. So we starting for the sampling, we have collected ovigerous female from Posidonia oceanica meadow of Lacua Ameno using a planktonic traveling net modified for the sampling of vaginal fauna of seagrass. These organisms were sorted on the boat, and ovigerous female of Hippolyte inermis were transferred to the laboratory and cultured in 2 liters conical flask until the larvae hatch. The larvae of Hippolyte inermis were reared in the thermostatic chamber in controlled condition with a temperature of 18 degrees and with a photo period simulating day and night in the conical flask of 800 millimeters with a larval density of 1 larvae for 10 millimeters. These larvae were fed with a mixture of enriched Artemia salina and rotifer Braconius plicatilis until the post-larval stage. So after about 30 days, these larvae reach to the post-larval stage. From that moment, post-larvae are collected and each batch divided into 2 groups. The first group is fed with a basic food consisting of spirulina and lyophilized Artemia, and this group is called PL5 manus. The other group is fed with the same basic food, but also with diatoms, coconeis, and this group is called PL5+. So we prepared for diatom treatment, so diatom fed on post-larvae, and for the base food treatment, so without diatom fed. 3 technical replicates of 70 biological replicates each. After 5 days of treatment, the post-larvae are fixed with a liquid nitrogen and stored in the freezer at minus 80 degrees. The arena was extracted from the whole sample and sent to the navogenes for the transcriptomic assembly. So we have more than 100,000 genes have been identified into Hippolyte inermis transcriptome. So now we have obtained a database of different expression of genes, of all genes. So more than 9,000 expression genes inside, depending on the treatment. We then made a heat map of these genes according to the expression. So now this map, the green colors corresponding to the high expression, and inversely for the red with a low expression. 2 clusters corresponding to the different treatment can be seen. One corresponding to the NFD, so without just only basic food and without diatoms, and another with corresponding to the FD, so basic food and diatoms. We notice a slight difference in ones of diatom-fed replicates. The diatom-fed number 3, so FD3, in which the gene expression in some cases is different if compared to the other 2 replicates of FD1 and FD2. Considering that the apoptosis of androgenic gland is a process that happened in a few days. Probably one of these 3 replicates were a slightly different time point of the process. So we created a second heat map with only genes with a p-value less than 0.0001. And we can see 2 other clusters, A and B, corresponding to the gene. The cluster A is a group together the gene with high expression with the treatment with diatoms and a low expression with the other treatment. And inversely for the cluster B. In addition, increasing to the stringency of differently expressed genes, smaller differences in gene expression can be seen and demonstrated in the quite perfect crystallization of treatments and replicates. So for confirm this result, we make a correspondent analysis. And looking on the first axis, we found our two previous clusters, so A and B, very clearly separated with the genes corresponding to the treatment. Base food on the right, and the gene corresponding to the treatment, diatom fed on the left. On the second axis, we can see that the group without diatoms is very compact and low distributed, at the opposite to the group with diatom fed, which are much more vertically dispersed. We can speculate that the different distribution in the relative to the presence of one replicate with slightly different patterns of gene expression. I said before, it can be correlated to the different time points in the apoptotic process of the various replicates of diatom treatment.
Thank you, Thomas.
I suggest to take away the mask while you speak.
Okay, sorry.
Thomas for the French, for the mask was not.
Can you hear me better now? Okay. So, thanks, Thomas. Using the tunnel method for the apoptosis detection, during the previous investigation, it was demonstrated that a known diatom-backed compound triggers specific apoptosis into the androgenic gland of decapod Hippolyte inermis. We can see in the picture a section C where the fluorescent tissues indicate the presence of apoptosis into the androgenic gland of the shrimp. We know that the regression of androgenic gland will affect the crustacean sex reversal. In fact, we know that the effect of compounds contained in the diatom of Cocconaeus genus is specifically targeted towards the androgenic gland, destroying it during the first post-larval development and causing the following regression of the male gonad. Female gonad is then produced starting from an undifferentiated cells only after the complete destruction of testes. For this reason, we search using KEGG analysis genes involved into the apoptotic pathway in arthropods, as well as other programmed cell death processes. We found several genes that are activated in post-larvae fed with the Cocconaeus diatom. And in particular, I am showing here just the main programmed cell death process. We found that GRIM, that is an activator of apoptosis, as well as caspase-9, and that is called Dohrn in this case, and caspase-3, that is called RICE. They are respectively the initial caspase and the executioner caspase. They are upregulated in post-larvae fed with diatom. Considering this previous paper, it is reasonable that this apoptotic process is part of this highly targeted destruction of the androgenic gland triggered by this still unknown compound produced by diatom, this diatom Cocconaeus. The upregulated K genes involving the programmed cell death are the same found by Nappo in 2012 in human breast cancer cells treated with Cocconaeus fraction. In addition, we found few genes involved in other programmed cell death, such as some in ferroptosis pathway or some in autophagy pathway. I didn't show in this presentation. We are still working, trying to reconstruct this pathway in crustaceans. In addition, we know that insulin-like peptide produced in the androgenic gland is fundamental for the sexual differentiation, and as we have seen in Heli presentation. It is involved in sexual differentiation, development of testings, maintenance of masculinity in crustaceans, and growth. Sidis group and Heli, they published many papers on these topics. So thanks to our Israeli collaborators. The production of insulin-like hormone is typical, as we have seen, of males individuals. So, using KEGG analysis, we found several genes involved in the pathway of insulin secretion that are downregulated in 5 days old post-larvae fed with diatom. I want to remember that to the audience that post-larvae fed with diatom will give sex-reversed female shrimp. These genes indicated by blue arrows are involved in the secretion of various insulin-like peptides, as well as they have been demonstrated to be involved in the activation on some white line cells called homocytes in invertebrates, in particular cell death processes and in some innate immune system reaction. If these results will be confirmed by real-time investigation, it could be the indication that sexual destiny of this species of shrimp appears soon after the feeding with coconase. So, how are results? First of all, performing KEGG analysis on the whole set of 100,000 genes, we identified 335 different pathways. Of these 100,000 genes, 9,000 of them are differently expressed in 5 days old post-larvae fed with diatom versus 5 days old post-larvae fed with only base food. These evidences showed, using, for example, correspondence analysis and EPAPs, demonstrated that for sure some biological process is occurring. And in addition, we found some phylogenetically conserved K genes involved in the programmed cell death that are upregulated in the diatom-fed post-larvae. And finally, there are several genes in the insulin secretion pathway that are downregulated in diatom-fed post-larvae. In conclusion, the presence of coconase diatom in shrimp food is directly correlated to the activation of various genes belonging to various pathways in this first day of Hippolyte inermis post-larva development. Our investigation demonstrated and confirmed that some programmed cell death process is activated by the diatom by active molecule. Finally, although the complete secretion pathway of insulin secretion is not fully discovered in crustaceans, some genes involved in this process are downregulated in the diatom-fed treatment. And this probably is connected with the hormone induction of sexual differentiation in these 5 days old post-larvae. As future perspectives, in the next month we have to identify if the COVID will begin, future genes pathway, validate, identify the differently expressed genes by real-time qPCR, and write manuscript, and identify demonstrate biotechnological application of our investigation in aquaculture practices. Thank you for your attention.
Okay, thank you, Mirko, for your great presentation. As we can see, and we actually recognize that the huge amount of data generated by the two transcriptomic libraries actually opens a huge amount of data mining and research, and we have a lot to do in the future. Hope that we'll have, of course, continuation of this great work. Amir, want to say something?
I think that those two lectures that we just saw are really showing the quality of this collaboration, because what you see here is really, on one hand, the differences between the stages in the life cycle of the this hermaphrodite shrimp, and in which a single specimen is crossing the borders between the different states of reproduction. This is very unusual in decapod crustaceans. You can find not so many such examples, and here in the example that we are demonstrating today, we have one, the natural occurrence of sex change on the one hand, that was shown by Ellie, and on the other hand, the enforced change by diatoms that was shown by Mirko. And together, I think when we are crossing the transcriptomic data from the two different lectures, there will be a lot to understand on the natural processes that are occurring over there. So there is, as I said before, there is a room, a very large room for future studies, and also this, up to this point, it will result quite fascinating data sets and publications.
Okay, thank you, Amir.
We have a question from the streaming for Heli. Which is the first biotechnological application of this project, of all these results, in your opinion?
Sorry, I didn't hear you well.
Roberta is asking us, is asking you, which are the first biotechnological application, in your opinion, of this project, of these results?
First of all, the understanding of a new candidate role, new candidate gene's role in the sexual differentiation process opens the way for a much diverse biotechnological processes that we can use, either in monitoring or influencing sexual characters, the visuality of individuals, like in terms of market. If you want to actually to influence morphotype, different morphotype, for example, or to find more elegant or more easier to do or to apply method for sex shift. At present, we are using biotechnologies using RNAi, but maybe with more knowledge on other genes, it will open the way for more simplest method to do it. So there's a lot of things that you can see that open when we are dealing with more and more genes that are related or actually could be manipulated in the process of sexual differentiation.
Okay. There is another question from Edward related about the connection of these studies on apoptotic processes with the fight of cancer. Valerio, I think it's if you want to.
Yes. Besides the aquacultural properties of these merits of this project, we can see also some application in medicine, because if we can be able to find, first of all, the compound, the active compound inducing apoptosis, and secondarily also the way it works, then we can have keys for producing new compounds that could be useful to produce new medicines. This I also would add some details on what Mirko was saying before, because Mirko, you showed this interesting pattern of induction of apoptosis, and you said it can be due to the apoptosis of the androgenic gland. Now we know that the AG goes under apoptosis according to this compound, to the administration of this compound, of this diet. But in my opinion, that is such a small gland, and it is such a fast process, because we know that it happens in 24 hours, that because in this stream, another peculiarity is that the apoptosis of the AG goes very fast in respect to other species, that we should be really very, very lucky to get that signal. But we also know that according to the apoptosis of the AG, we have a cascade apoptosis of the vas deferens, testes and other tissues. And probably the signal that we are reading is that. We will have time to discuss about this, but I think this shows how the process is still under research, and it's so exciting to try to put hands in this process and understand how it works.
Okay.
Okay, thank you, Valerio. Enrico, are there any more questions regarding the? Okay.
Yes, there is another question here. About the compound produced by Dalton, if we know something about this compound and how to identify it. This is a apoptotic compound.
Okay, this again goes to Valerio.
I think
it's a good question. The compound is still unknown, and the reason it is still unknown is because it works at very low concentrations. It's almost homeopathic. So we are working with different research groups of chemistry, and they have big difficulties finding this compound because it is hindered by a lot of more abundant compounds. At present, we know only that it's a very small compound, very lipophilic. It is around 21 atoms of carbon, but we still ignore its structure because it's a new compound, not known for science.
Okay.
That's all.
Thank you.
Okay, it's 11. I want to give my personal compliment to all the speakers of yesterday and of today to be always very sharp on time. I've never seen a congress in presence where timing was so well respected. So I suggest to go to take our coffee and be back for the round table reserved to scientists and of all the projects at 11:30, and then see back in streaming at 12:30 for the conclusive remarks at the end of the conference.
Okay. Thank you all.
Good coffee.
Yeah.
Valerio.
Sì.
I'm sorry, but thanks for the invitation to join the roundtable. But unfortunately, I have India waiting for me in half an hour. Today I'm in the office. So if you agree, first my compliment.